talk is entitled Functional Ultrasound Imaging.
Well, thank you very much. Sorry about that. Never should bring a new PC. But it's a great pleasure to be here. Thanks for the invitation. I'm going to talk about functional ultrasound, and I'm going to talk about what we can do today, and then in terms of blood flow, and then I'm going to talk about can we measure the true blood velocity today. I'll look at what we can do today, and then what we currently can do. And if we can measure the velocity vector, can we also measure pressure? Can we measure the pressure in the vessels? And then I'll take a little departure from that and going into synthetic aperture imaging, which is a new method of doing ultrasound, which also has better focusing properties. And then I'll look at how we can do flow with that and how we can do fast vector flow. And then I'll conclude by looking at can we actually measure the full three-dimensional velocity vector and look at that. But first of all, let's try and look at a conventional ultrasound color flow image. What we're seeing here is an instantaneous display of the velocity in the carotid artery. So what we're seeing is that the red color to us indicates that the velocity is going towards the transducer. There are, however, a few problems here, because you can see that the peak velocity here is around 28 centimeters a second, but if we look at the peak velocity in the carotid artery, it's around a meter per second. The reason is that we are only looking at the component going towards or away from the transducer. So we're actually not seeing the whole picture. Here it looks very nice. This is what the manufacturers like to show you. And here is what they don't like to show you. This is when we have the beam to flow angle at a very poor angle. So now it's going transverse. And now you can see at the one part of the vessel it's going in one direction, and the other part of the vessel is going in the other direction. Of course, quite clearly flow doesn't go like this. So what is happening here is that we have this angle. So what we're seeing is the true velocity times cosine to the angle. And if you can see, the further you go down to 90 degrees, the less you see. So in this case, we were around 90 or 80 degrees, we only see 17% of the velocity. That's why when we look at the scale, it says 8 centimeters a second, where it should be 1 meter per second. So of course it's a fundamental problem in the system that we have today, in that we don't see the full picture. In this way, in this example, it's not so difficult. We can just angulate the transducer or something. This is very simple. But look at this. This is if we go slightly up the carotid artery and we go to the bifurcation. And when we're looking at the bifurcation, where's the flow going? What's the velocity? It's essentially just a jumble of colors. So we can't really see something. From this, and I'll show you also later, it's very difficult also to correct this. Because in the previous example, we could just say, okay, the angle is 80 degrees. We compensate for that and get back to the correct velocity. But what is the angle here? It's quite obvious that there is not one angle. You cannot correct this. You should correct this for all places in the image 20 times a second. That's not a thing you can use in the clinic. So what we do need here is the full velocity vector. So let me come with a little bit of background on what we're measuring today. If we're looking at it, the orange-yellow thing is the ultrasound field. What we're looking at, if we go right down, we see an oscillation. And we can see that we have a response when the scatter crosses the field going downwards of a sinusoidal oscillation. This sinusoidal oscillation, when the scatter crosses the field, we can detect. So what we do in these kind of systems is we detect that kind of frequency we have there. But then when we go across the field, we can see that we don't really get a frequency. If we go on the diagonal, it's okay, it's down in the corner. But when we go across, we have no frequency. That's why we don't detect anything. So the basic idea in what I'm suggesting here, why don't we make an ultrasound field where we have an oscillation across? So when we go across in direction there, you can see we get a lateral response on the right, which has a frequency. We can detect that kind of frequency. The question then is, can you generate fields like that? And you actually can. You can sit down in the bathtub, you can put your 2 fingers in the bathtub, take them up and down, you will see 2 spherical waves going out. And when they intersect, you will also have a transverse wave. So what is happening here is what you need to do is to split the transducer and make 2 places where you send out sound from. And when you have 2 places where you send out sound, then they will interfere, and you can see you get this pattern where we, going from left to right, have the normal axial oscillation, and going top to bottom, we have the lateral oscillation. What we essentially do when we implement this in the scanner is that we send out a normal ultrasound field, and then we create everything in receive. So we have 2 receiving apertures. It's a detail, but we can generate fields like this. This is what you can see. So this we implemented in a research scanner, and here you see one of the first in vivo examples of it. On the right side, you have the conventional image like you would see in a normal scanner. And note here the very dark colors, and that we're in this region of 0.1 meters per second. On the left, you can see that at the lower, we have the carotid artery going from left to right. You can see the arrows. The colors also indicate velocity. And on the top, we have the jugular vein with a return flow from the brain. So you can see that we now start to get the right dynamics, and we get up in the velocities that are physiologically correct. Then here is the example from before, and now it starts to get a little complicated. So we moved up the carotid artery, and we're now in the bifurcation. And what you can see here is we still get that jumble of colors on the right. And then on the left, you can see the arrows are pointing in all sorts of directions. It's quite difficult to see because there's so much information there. So let me take a small freeze frame here. This is how it looked on the conventional system. And then if we switch over to the vector flow, I think we can see what's happening. What is happening is that the blood is coming in on the left, and then it hits the flow divider, and then it gets bent down, and then we have a vortex, which is forming down and going out. So you see you have a fairly laminar flow on the top, and you have the vortex on the lower branch. And the problem of looking at this is really that this vortex comes in the systolic phase. It appears in 100 milliseconds, and then it races down the stream at 1 meters per second. So it's in the view for a fairly short time, and that's why it gets to be complicated to look at. But now you can actually start to see what's going on. You can see that we actually, in this case, have velocities going in all directions, 360 degrees. And the velocity direction and the magnitude changes on these timescales that we are looking at here. This has been implemented in a commercial platform here. It was FDA approved in January. It's now in the market. And they have introduced the color wheel. So now you have to get used to that the different colors indicate different directions in the image. And if we take the example here, we're in the carotid artery, and you can see now it's fairly laminar. And now we're going upstream, so we're going up to the bifurcation. And what you can see in the top branch is that a mixing of other colors comes in, and that's because of this vortex. So we have that at certain parts in the cardiac cycle, we can see green and red, so it's tumbling around. And this is what we see in normal patients. So this is what happens for any kind of human being. And the interesting thing is that you see it in the carotid artery, and other arteries you don't.
Just a quick question. Maybe we'll get to this, but
what's your confirmation that this is accurate? I mean, that this vortex is forming? Is there some independent metric by which you can say, yes, exactly?
Well, the thing is that you can also measure this vortex with MR scans. This takes you half an hour. The way we validated this was with MR scans in the carotid artery, looking at stroke volume. So there was more than a 90% correlation between the stroke volume of MR and this. So of course, you're right. It's really difficult to say what's actually going on. Let me take you to another complicated example. This is where we have scanned a patient in open heart surgery. So we are looking at the right atrium. And what you can see here also is how the complicated pattern is that the flow is rotating around 360 degrees. This is very early data, but what we of course hope to do with this is that when people get a valve replacement and things like that, it's actually possible to go in the operating theater and look at this and see that the flow is actually working as it should before they close the patient. One thing that's interesting with having complete flow data is that we can also use this for finding the pressure in the vessels. So here we've taken a carotid phantom with a 70-degree constriction and then measured in this. And if you look at this, then we measured at the red spot, and the top graph, the black graph, is the velocity, and the red one is the derived pressure. So from solving Navier-Stokes equations and looking at the vector flow data, we can actually start looking at the pressure gradients and the pressure in the heart and around the valves and things like that. So this was sort of the first part of it. Now I want to go to another place where we say, let's not look at the conventional thing where we go in a serial fashion in the same direction. Now I want to introduce synthetic aperture. In synthetic aperture, we use a small aperture and then we send out sound in all directions. And when we send out the sound in all directions and receive with all channels, we can actually formulate one image from one shot. And then we go over the apertures as I've shown you on top, and then we acquire with one image, at the whole aperture, but send out with a part of it. And when we've collected all of that data, we can actually make an image which is perfectly focused in both transmit and receive, because we have the full data set. And you can actually go in the clinic and you can compare with normal imaging and see that that actually translates into better images. But what I want to concentrate on today is flow. And of course, it's a somewhat ridiculous idea to spread out your imaging over multiple shots when things are moving. So the question is, can you actually make flow with this? The reason why I'm so interested in this is that if you can insonify the whole region and look at the whole region, then I can continuously monitor the velocity in the whole region, which would give me the possibility of averaging over a long array of shots to give a really good flow image. And also, if I have a short sequence, I can make it really fast. The trick here, of course, we thought about this long, and of course the trick to solve the problem is to ignore it. Because you can find the velocity of a car even though you take it on a long exposure time. Just if you take two images at a long exposure time the same way and then compare the two. Yes, they're blurred, but they're blurred the same way. And that's what we're doing here. So we're just taking a short sequence, reconstructing the image, and then we're just comparing it. And if we compare the same imaging sequence, then we can actually get a velocity out of it. And now we're looking in all directions at the same time. If we're looking at, this is the first in vivo image we did, and if you take a normal scanner and do an image like this, you have to shoot 800 times or something. This is done in 24 emissions. So it's over 20 to 30 times faster than a normal color flow image. That means that you can make 900 images a second down to 4 centimeters, or 200 images of the heart down to 15 centimeters. So you can see the complex flow with the vortices and things going around, and now you can study it in really good temporal detail. Here is an example where we have a frame rate of 100 Hertz. I've slowed it down. Here comes the systole. You can see the strong acceleration, and then it dies down. So you can start seeing these kind of things with a good temporal resolution. And I think that's important for valves and vortices and all these kind of complicated things that we see in the body. Another benefit of this, when we're looking at an image where we're continuously looking in all directions, is that we can average over a long period of time. That means that we can also see very low velocity flow. This is just an example taken from the literature using a contrast agent for a bladder tumor. And what is done here is that the vasculature of the tumor, of course, is really, really important, but you have to use a contrast agent these days to find this. But I think that if we have continuous imaging, and if we can average over a long period of time for the slow velocity, we would also be able to see some of that. Bigutander is here, he's going to show some of it also later on. Another thing that we've been working with has been the fast plane wave imaging. You can send out spherical waves, but you can also send out plane waves. What is done here is that we're sending out a plane wave that insonifies the whole region, and then we reconstruct the image from that plane wave. And the nice thing about it is that you get one image every time you send out one frame. So you get incredibly fast images. You can get 5,000 images a second. If you want to do flow, you have to combine 10 or 20, but you can get hundreds to thousands of flow images a second. This is an example of a jugular vein and a carotid artery. On the top, you see the vein, and on the bottom, you see the artery. And now it gets really confusing, because why do we have all this pulsation in the vein? And again, I have to freeze things a little bit. The thing we're looking at is a valve. It's actually an incompetent valve in the jugular vein. So what you're seeing is, on the first image, that we're having a flow going from your right to left into, and you can see the vortex behind the valve goes in the clockwise direction. But this is an incompetent valve, so it doesn't close this, so the flow is going back again. And that's when you see the flow vectors going in the other direction and the vortex changing sign, so it's going counterclockwise. So of course you can start seeing this kind, does the valve work or not? One interesting by-effect we saw in the carotid artery is that here you see the flow going directly out, but you can also see there's a rotation. So it does not, it also rotates around, so it makes a vortex in this direction, but it also rotates around in this direction. So when we look at it, we have motions in two different planes. So what we really should do in order to see the full image of this is to do it in 3 dimensions. So actually see the 3-dimensional velocity vector. So to do this, we have to be able to control the beam in 2 directions. So we built Saurus. So we can now put on a 32 by 32 array transducer on this. So that's 1,000 channels. So this samples at 70 megahertz, so we generate 140 gigabytes of data a second in this. There's enough memory in this for storing 3 seconds of data. And then it can also do real-time processing on it, loads of software, all sorts of funny things. It took us 7 years to build, but now the toy's there, so we're really happy about it. So we made an example here with a Vermont 32 by 32 array transducer. And here you're seeing now we're cutting the vessel straight across. So in the normal imaging plane, we shouldn't see anything. It's only a motion going through that plane. And this is how it looks. So here you can see Vx is the transverse direction, Vz is the depth direction, and Vy is the direction going in and out of the plane. And by using a 2-dimensional array, you can actually see all 3 velocity components, and then you can see that you have a fairly round flow in this. So this is sort of my conclusion on functional ultrasound imaging. I think you can start seeing functions, you can see vortices, you can have fast imaging, so you can study things at a high temporal resolution. I think it's possible to look at perfusion imaging, and I think it will have an effect in tumor. It's probably possible also to get pressure estimations to see the pressure distribution in vessels in the heart. and I think we will be able to do it in 3D. So thank you.
Jørgen, you say that you should be able from the velocity to go back to pressure.
Yeah.
You know that there is mixing in pressure, velocity, and sometimes elasticities, arterial elasticity. But here, as you've got all the vector flow, all this information, perhaps just by checking the way your vortices are going to be created, you could be able to have access to pressure without being corrupted by arterial elasticity.
Yeah, I think what you can do is you can look at the velocity vector in a small region around the point, and then derive the pressure, and derive the pressure gradient, and then assume, okay, there's a place where there's zero pressure, integrate out and find the pressure. So I think you should be able to do that, and I think if you're looking at how valves or things like that you put in, you would like actually to see the pressure gradient and the pressure distribution.
First, did you try to find a viscosity also?
We did a small experiment where we looked at the influence of the viscosity, and you have to be so heavily doped that you're dead before it has an influence. So.
Coming back to this question, I can imagine that you get the transient part of the pressure field, velocity measurement. But how would you get the static part?
You don't get the static part. This is sort of, you say, it is the pressure gradient. It's related to the pressure gradient. So it's not the absolute pressure. But if you measure with the pressure curve, I think you can calculate that. And I don't think that the static pressure as such is the most important for the dynamics.
Other questions? Thanks, Jørgen.